Hello, welcome to my lecture on Chapter 11. Chapter 11 is all about how the expression of genes is controlled at the cellular level. And we have all of these genes in the genome of a given organism, and in eukaryotic organisms there are different cell types, and the cells, depending on what parts of the genome are expressed, produce different proteins. Okay, so we have already talked about how DNA is replicated and how DNA is transcribed into messenger RNA, and then how messenger RNA is then translated into protein. So we know how the genetic information flows from gene to protein, and that overall process is called gene expression, how the genotype is expressed into the phenotype. Gene regulation is how certain genes are turned on and off, and that can help organisms to respond to environmental changes. And I talked a little bit about gene regulation when we were covering Chapter 10, but it's a little bit different in prokaryotes versus eukaryotes. And in prokaryotes, there are sets of proteins or sets of enzymes that are controlled together, and those units or sets of enzymes and proteins are called operons. And there are regulatory proteins that bind to particular sequences in the DNA that are control sequences and determine which genes are expressed. So here is an example of that in prokaryotes. There is a lactose operon, and so lactose is a protein, and if there is no lactose present, then the operon, or set of enzymes responsible for breaking it down or digesting it, is turned off. If there is lactose present, then the operon is turned on. And so this particular operon is regulated by a repressor. So if there's no lactose, then the repressor is activated and turns off the operon, and if there's lactose present, then the operon is turned on, and the enzymes for breaking down and using lactose are produced. Okay, this is just So there are different types of regulation mechanisms for operons in prokaryotes. The lactose, or LAC operon, is what's called inducible, and then this is TRP, or tryptophan operon, is repressible. An inducible operon is one that is turned off unless the substrate is present. And then if the substrate is present, then the repressor becomes inactive, and those enzymes that break it down and use it are produced. The tryptophan operon is an example of a repressible operon, and so in a repressible operon, if the substrate is present, then the repressor is activated, and if there is no substrate, in this case tryptophan, present, then the repressor is inactivated, and the enzymes responsible for breaking down and using the substrate are produced. Okay, so here's a really good checkpoint question for you to answer on your own. A certain mutation in E. coli impairs the ability of the Lac repressor to bind to the Lac operator. How would this affect the cell? the way it would affect the cell is that even if there was no lactose present, the repressor would not be able to bind the operator region, so the pathway that produces, or the operon that produces the enzymes that break down and utilize lactose would still go, and so you would have an excess of these lac operon enzymes present. Now we're going to talk about chromosome structure and different things that can affect gene expression in multicellular eukaryotes. As a zygote, or the first cell of a eukaryotic organism, develops into a multicellular organism, there are different types of cells that are needed to form that multicellular organism. So the cells undergo what's called differentiation to become specialized in their structure and function to meet the needs of the organism. And so each of the cell types has a specific set of genes that are expressed, while meaning that some genes are expressed and others are not. And so depending on the cell type, you have, some proteins produced while others are not. And selective gene expression accounts for the difference in which proteins are produced in each specific cell type, rather than, different genes being present. The same genes are present, they're just expressed selectively or differently depending on cell type. So even the structure of a chromosome can affect gene expression. For example, we have talked about the structure of DNA and the fact that DNA strands are very, very long, and that DNA is wound around small proteins called histones, and they form a bead-like string of nucleosomes. And so DNA packing, or that winding of the DNA around the histones that allows it to fit into the nucleus, tends to have the effect of blocking gene expression by making it impossible for transcription to proteins to bind DNA. One example of DNA packing is called X chromosome inactivation, and that happens in female mammal cells. Chemical modification of DNA bases, or the proteins, the histone proteins, can also result in epigenetic inheritance, which is just when the inheritance of traits are transmitted by mechanisms that are not directly involved with the nucleotide sequence. That's epigenetic inheritance. Okay, so here is another really good checkpoint question. In your body, a nerve cell has a very different structure and performs very different functions from a skin cell. Because the two cell types have the same genes, how can the cells be so different? The answer is because the gene expression is different. Not because the genes present are different, but the expression of the genes in the different cell types. In order for transcription to happen, there are several steps that allow for RNA polymerase to properly attach to the promoter region in the DNA sequence and begin transcription. One of the things that needs to happen is a protein or proteins called transcription factors need to assist this whole process. The transcription factor has to bind to DNA control sequences called enhancers that are usually located kind of far away from the gene they regulate on the DNA or on the chromosome. And so then a DNA bending protein twists the DNA strand and brings the bound protein activators close to the promoter. And once the DNA is bent, then the activators are able to interact with other transcription factor proteins, which binds as a complex or as a group at the gene's promoter. And then, and only then, can RNA polymerase attach to the promoter and start transcription. So for this checkpoint question, what must occur before RNA polymerase can bind to a promoter and transcribe a specific eukaryotic gene? The answer would be that the transcription factor proteins need to bind to the DNA control sequences or enhancers and then interact with other proteins, regulation proteins, in order to bind as a complex at the gene's promoter, which then allows RNA polymerase to bind. Then once transcription is completed, RNA splicing further impacts gene expression. And we talked about RNA splicing. It happens after transcription, and two or more types of messenger RNA can be generated from the same transcript or the same sequence of DNA that was transcribed into mRNA. And in humans, over 90% of genes that code for proteins undergo alternate splicing. So, in other words, can be expressed differently due to RNA splicing, which leads to this checkpoint question. How is it possible that just under 21,000 human genes can produce over 100,000 polypeptides? Well, the answer is the regulation of gene expression. You have differences in, how genes are transcribed, and then after transcription, you have differences in the way that the mRNA sequences are spliced, which can lead to different expression of genes or the differentiation in expression of proteins from one cell type to another. In other words, in different cell types and different mRNA sequences, you have, different introns, which are the ones that are not expressed or not translated into proteins, and different exons, which are the sequences of DNA that are expressed or that are translated into proteins, resulting in different mRNA transcripts that end up in different cell types, or what I should say, end up being expressed in different cell types. In addition, the later stages of gene expression can also be regulated by different factors. For example, mRNA molecules can have different longevity, meaning that some are longer lived than others. So if you have a long-lived mRNA sequence, then that can produce a lot more protein molecules than a shorter-lived mRNA sequence. Another way that gene expression is regulated is at the translation stage, and the process of translating mRNA into a polypeptide or protein has different ways that it can be regulated. There are a lot of proteins that control the beginning of protein synthesis or polypeptide synthesis. Some of them control the translation of all mRNA sequences, and other regulatory proteins, when it comes to translation, are specific to cell type. For example, red blood cells have an inhibitory protein that does not allow translation to happen for the hemoglobin molecule mRNA unless the cell has a supply of heme, which is the iron-containing chemical group that is necessary for hemoglobin to function. And finally, even after translation, protein processing can impact gene expression. So a lot of times those polypeptides that are produced during translation require some processing before they can become functional. One example is that in insulin, the cutting out of a section of the original polypeptide that's translated is needed in order for that hormone to be active.